Now, I've spent a lot of time talking about primary visual cortex, possibly giving you the impression that this is the place where vision happens in the brain. But really that's just the beginning. There's a plethora of other visual areas which are involved in achieving our visual perception. And in fact, people say in humans around a third or even a half of the brain is involved in vision in different ways. So vision is such an important sense for us humans. And this diagram just shows a few areas that have been identified, and I'm just going to talk a little bit about one of them because this is particularly well understood and seems to have a particularly distinctive role. MT, sometimes known as V5, seems to be a cortical area which is particularly important for the perception of motion. So we saw that V1 contained some direction selective neurons, but they're a minority in primate V1. Most primate V1 neurons are not tuned to direction, but those that do project to this cortical area MT. It's called MT because it stands for medial temporal area. It's in the middle of your temporal cortex. It's kind of handy because MT looks a little bit like an acronym for motion. Now in MT, almost all neurons are strongly direction tuned, so that's a bit of a clue that it's involved in motion perception. Here is an fMRI image showing the results of a stimulus that's designed to detect where in this person's brain MT is. You can't be exactly accurate and say we know exactly where MT is, so they talk about the human MT plus area. So that's thought to be equivalent to monkey MT plus a little bit extra, some other areas that respond to motion. And to get this area of the brain, they had the person view expanding dot patterns and contrasted that to the brain activation when the person was viewing static random dots. And so they're picking up the brain area that responds much better to these expanding star field pattern of dots than to the static dot pattern. And that very typically reveals this little area here, as you can see, sort of in the middle of your temporal cortex. Now this is a figure from a nice review of MT, which is on Blackboard for you. And you can see that MT receives strong input from V1, particularly from cells in V1 layer 4B. And you can see it also gets inputs from V2 and from V3, and interestingly from the coniocellular layer of the thalamus directly, and from some other pathways that we haven't even talked about yet. So you can see that from the retina, some of these retinal ganglion cells, the W retinal ganglion cells, project to the superior colliculus, and then via the pulvinar, another area of thalamus, project on to MT. So you don't need to remember all the details of this, but it's just interesting to see the range of visual areas from which MT is receiving input, just giving a sense of how complicated this circuitry is. We discussed orientation columns in V1, while actually several other cortical areas have a columnar architecture. This is a nice figure from a paper by De Angelis and Newsom showing that in MT you not only have direction columns, which are kind of equivalent to orientation columns that you find in V1, you also find disparity columns. So you remember we talked about stereoscopic disparity, the small offset between a stimulus position in left and right eyes. Well, here they've colour-coded cells that they found that were tuned to particular disparities. In red, to disparities indicating an object closer than the fixation point where the monkey was looking. 0 cells that are tuned to a disparity at that fixation point, and green cells that are tuned to a disparity further away. As you can see, you're not encountering that tuning at random with MT. There's a smooth progression from near to 0 to far among the subset of cells that are disparity selective. So once again, you've got two maps kind of superimposed on top of one another here. We don't quite know the significance of this columnar architecture yet, but we imagine it has something to do with the computations that are carried out by these areas of cortex, and presumably it's to do with minimizing wiring. So we assume that in the process of vision, the brain more often needs to compare inputs from cells that are tuned to similar directions and to similar disparities, and so it's going to be convenient to have those cells nearby one another so that you're not sending very long axons all across cortex trying to do these basic computations. A clue as to MT's distinction role in motion perception is given by the effects of lesions to MT, which can occasionally occur after brain injury such as stroke or in an accident. And MT lesions are associated with a very strange visual impairment called akinetopsia. So that's the inability to perceive motion. It's sometimes called motion blindness. And an example patient who was described in the literature who suffered from this described her difficulties. She found pouring a cup of tea or coffee difficult because she couldn't see the fluid moving up the cup. One moment the tea would be at the bottom of the cup and then the next moment it would be overflowing. Again, people, they sort of appeared and disappeared, but she didn't see them move from one place to another. So you can imagine how dangerous it was to cross the street. And there's a link here to a video giving some more examples of the difficulties of people with akinetopsia.